I'm enjoying grading the tests. Well, it's ongoing. I hope to have it done by the end of the week. I typically do not post an answer key until I'm done grading. And that way, if I see anything that needs adjusting, I can include it in the answer key to get the information. And so this is a really good opportunity for you to go over your test and work the problems that you try to get trouble with, or talk about what was going on. Everybody's taking it, so when we have questions from office hours, we're looking at each other. We have the temptation of looking up the answer and just automatically seeing, oh yeah, I understand that. Okay. Again, never missing for a learning moment. All right, so moving on. Now we're going to start talking about a really good idea, understanding of the structure of the genome, DNA sequence, how it relates to the gene, and expression of the gene, transcription, and possibly translation. We have a really good idea of what a gene is and how it makes its product. So we're going to start talking about alterations in that genome, alterations in that DNA. Now let's start right off here. The DNA together. So we'll come back to this topic a little later, but I want to focus on this, which was the most common wrong answer when you answered this question in one of the first warm-ups at the beginning of the semester. And that many of you believe that it couldn't be mutable. In fact, it has to be mutable for evolution to happen. That genomes have to have some flexibility, have to have some degree of instability in order for change to happen, so that in case there is a change that is adaptive, is beneficial, it can be acted on through evolution and fixed in the genome and so on. So there has to be some level of instability. So this balance here between having mutable genome and yet it can't be so mutable that it leads to chaos every cell generation. So we're going to talk about that mutability. And by way of defining different types of changes to the DNA, different mutations, a mutation is nothing more than a change in the DNA sequence, a change in the nucleotide sequence that alters what the most common form is, or what the original version was that before that change happens. So we're going to talk about different kinds of changes. Your DNA is basically the target for assault from all sorts of areas that can lead to changes in the sequence. Replication errors. We talked about replication and how this is really can be a pretty error-prone process. One of the reasons that it's less error-producing than it could be is that there are repair processes in place to fix many of the changes that happen spontaneously just by mistake. Not all of them, but some of them. And we'll come back and talk about those repair processes after we talk about mutations and the origins of mutations. So those replication errors can lead to gains or losses, additions or deletions of nucleotides. It can lead to incorporation of incorrect nucleotides, right? A missense mutation, a change, a mistake in incorporation of an incoming nucleotide can result in an alteration of that base pair at that position. Structural changes can happen. If some damage occurs to the base, the actual base attached to the sugar as part of the nucleotide, such that that base is lost, the adenine, the cytosine is lost, that structural change can lead to instability of that polynucleotide strand and can lead to an alteration in that DNA molecule. Heat can lead to base loss, and so one of the sources of that base loss is in fact heat attack on the DNA. You should be marginally familiar with the radiation and the different kinds of radiation. UV irradiation can lead to chemical cross-linking. We'll talk about that when we talk about induced mutations. Just very briefly, your text covers it. And ionizing radiation, which can result in breaks to the DNA, either a single strand or a double strand break. Obviously, if there's a double strand break, that seriously disrupts the integrity of that molecule. Each of our chromosomes is a single double-stranded molecule of DNA. So if there's a double-strand break, it's basically cutting that molecule into two pieces. That can cause a problem. Single-strand breaks can cause similar problems if they're not repaired. So these kinds of changes or effects on the DNA can lead to either repairs to fix them and they're inconsequential, or if they're missed by the repair processes, permanent DNA nucleotide damage or change in nucleotide sequence. So I want to talk first about, we'll focus primarily on the most common type of mutation that we will often encounter, and those are point mutations. And mutations that happen at a particular site. In its strictest definition, it's affecting one base pair, but sometimes we'll use point mutation to refer to two base pair insertion or two base pair deletion because it's a small change at one site. But if you look up point mutation, the definition will probably be alteration of a single base pair. Two basic types of point mutations: the substitution of one base pair for another. So we start out with an AT at one particular site, and a substitution would be a GC. Right? It's a change. It's an exchange, substitution of one for another. And the other type of point mutation is an addition or a deletion. Most commonly here of a nucleotide pair, we'll see more than that being changed. But inserting a base pair where in between two existing base pairs, we're deleting a base pair from a sequence. So these are two possible types of spontaneous point mutations. We're going to talk first about how these mutations of various kinds arise in DNA in what we call spontaneously, namely we don't see any obvious cause other than just a spontaneous mistake. But as you'll see when we talk about induced mutations, really that's nothing more than increasing the exposure to something that we're already exposed to that is producing spontaneous mutations, like radiation. Okay. All right, so what are these point mutations? There's some terminology to recognize. If we're looking at base substitutions, then it depends whether there is an impact of that substitution on the cell, the tissue, the organism in terms of phenotype or function. It's going to depend on what that substitution is, what kind of substitution it is. If there's a single nucleotide change that happens to take place in a position in a gene that is the 3' third position in an amino acid codon, where there's no change in the amino acid that would be directed during translation, it's technically a mutation because it's a base pair change, but we refer to it as a synonymous or a silent mutation. We wouldn't even know it's there because the same exact amino acid is being incorporated into that peptide chain during translation. But technically, it's a mutation because it has changed a nucleotide nucleotide at that site to a different nucleotide, causing that through replication, causing that to be a base pair substitution. More often we recognize the presence of base substitutions because they have an impact on function or phenotype. And that's going to depend also on what kind of a substitution it is. Less likely to have a significant impact on phenotype if that substitution of the amino acid, the base pair, results in an amino acid change that is an amino acid, the new amino acid is, chemically really pretty similar to the original amino acid that was at that site. That the chemical properties are pretty conservative. And so that's often what we would refer to as a neutral change. May have no impact on function or phenotype at all. Get a little bit more likely to have an impact on the function of that polypeptide if the amino acid that results from a base substitution is very different chemically from the original amino acid, or if that amino acid is located in a part of the protein that is absolutely critical to that protein's function. It might be a site, it might be a position in the active site of an enzyme where it wouldn't make any difference what amino acid was replaced, but that amino acid substitution might impact that polypeptide. Or it might just simply be that it needed, that active site needed a particular chemical nature of the amino acid, in which case a non-conservative missense substituting one amino acid for another would result in a functional impact or a phenotypic impact. And then if there is a change in a nucleotide that converts an amino acid codon in a protein coding region to a stop codon, that's what's referred to as a nonsense mutation. Nonsense mutation only makes sense in the context of a protein coding region, just as missense mutation only makes sense in the context of an amino acid encoding region of the gene. If we're talking about ribosomal RNAs, it makes no sense to talk about nonsense mutations. It's simply the consequence of having that nucleotide change on what the follow-up of that gene function is. And so having a nonsense mutation, depending on where that nonsense mutation occurs, if it happens very early in the protein coding region, it can result in a severely truncated polypeptide that has a high likelihood of having its function disrupted. If it happens very late in the polypeptide, it will depend on what that polypeptide is and whether it needs to have all of its amino acids right down to its normal carboxy terminal end. So it really depends. And there are in fact nonsense mutations that are associated with some disease genes that are less disrupted when they're part of when they're at the N terminal than they are, when they're down at the C terminal than they are at the N terminal because of the way the protein in quaternary structure is assembled. So the impact for these kinds of mutations on function and phenotype is dependent upon the specific location and product and function of that gene. Okay, but that's the terminology. And then we've got insertions and deletions, and these are often referred to as indels. So collectively, if I want to talk about the kind of change to the nucleotide sequence that is either the result of adding a nucleotide pair or deleting a nucleotide pair, collectively I might talk about them as indels. It's very interesting work a number of years ago that studied the DNA sequence for a large number of identical twins. Of course, we all think immediately identical twins, they're monozygotic twins, they start out with exactly the same genome. So if anybody's going to have exactly the same DNA sequence, it's going to be identical twins. It turns out that they don't, that there are a lot of small differences between the genomes of identical twins that have happened in the process of development, and many of those are indels. A random nucleotide insertion here, a random nucleotide deletion there, and they're different for the two because they're not sharing the development. They only share the starting nucleotide sequence, and that nucleotide sequence is behaving differently in the two with respect to where those indels happen. As long as they don't happen in functional genes, there are not any differences. But a lot of these changes in the identical twin genomes, as well as regulatory changes, can help explain why identical twins rarely look absolutely 100% identical and in every way. deleted, if it's in a region that is part of the protein coding region of a gene, it's obviously going to cause a change in the reading frame. And so often these mutations are referred to as frame shift mutations. If you insert a nucleotide in the promoter or a nucleotide in the 3' UTR, it's not a frame shift. If it's not changing the reading frame, it's not a frame shift. But people often will generally talk about frame shift mutations as generally insertions, deletions. Right? I'd rather make sure that you understand that if not every insertion deletion is a frame shift mutation. It is an insertion mutation, deletion mutation. If it's in a reading frame, it can be a frame shift if it's 1 or 2 nucleotide insertion deletion. Okay. So thinking about this, then think now if you know and understand the structure of the gene and the transcription and subsequent translation if it's protein coding, think about the molecular ability to recognize these kinds of changes or not, experimentally, molecularly, diagnostically. So we might think about these kinds of changes, missense mutations, nonsense, frame shift, regulatory, in the context of how we would look at the RNA product of the gene or the protein product of the gene. So here's just a generic example. We've got a wild-type gene that makes an RNA that is a certain length, and if you run a northern blot and analyze that northern blot for that one particular transcript, it has this size, so it's located here on the gel. And if it's a protein coding gene, we can also do the same thing with that cell's protein by running it on a Western gel and looking at electrophoretically where that moves, that protein, that polypeptide moves on the Western blot is, in the simplest analysis, is related to its size. And so we're looking at a protein that's this big coming from an RNA that's that big. If there's a missense mutation that changes a nucleotide, say at this position, from a GC to an AT, you're not going to see that on a northern blot. If that is part of what's transcribed, a nucleotide change is not going to show up as a difference in the northern blot because it's not changing the length or the weight of that RNA. It'll look the same. So we wouldn't be able to detect it that way. And even if that change is a missense that alters an amino acid, again, the simplest Western blot analysis is just simply primarily looking at charge and weight. We'll see weight, and so we're looking at the size. You're not going to be able to see that from a single amino acid chain either. There are ways to do this with the protein, but they're more complicated. But if we look at the consequences of a nonsense mutation, that is a change from a protein amino acid codon to a nonsense codon, again, no change in the RNA, we wouldn't be able to recognize that, but it could have a significant impact on the protein as we see it, as we analyze it in the Western blot. If it's a nonsense mutation that is happening upstream of where the normal stop codon is, it's going to make the polypeptide shorter because if it's a premature stop codon, it's terminating translation before it should, and so that protein is going to run, for that polypeptide is going to run further in the gel because it's smaller and lighter and run faster. Frame shift mutations, can't predict those because it's going to depend completely on what kind of impact that frame shift is happening. Is it going to cause does that frame shift introduce a premature stop codon? Does it extend the reading frame to a stop codon that's further on? And so it could easily impact the protein here. Again, you won't see that on the northern blot. And since we'll start eventually talking about regulating genes, but you know about regulatory sequences that start transcription, what if there were mutations in the essential, conserved, really important sequences in the promoter that eliminate promoter or transcription factor binding? In that case, we would see no RNA on that gel. Okay? In many cases, it might be something less than the abundance or more than the abundance if it's altering the transcriptional rate, altering the RNA rate. So think about this not only in terms of what those changes are, but what the impact of those changes would be on the products with RNA or protein, and how you would view or examine or detect those differences. If they wouldn't even know that they exist unless they have a phenotypic effect, unless you're screening everybody just randomly by nucleotide sequence. Okay. All right, so let's first talk about substitutes. Where do these come from? I'm going to talk about the origins of these mutations, and we'll start by talking about spontaneous some of the agents that can induce these mutations, basically by increasing the frequency of these errors. So the first one is substitutions of one base for another. There are two types of base substitutions that we can talk about: transversions and transitions. A transition is a change of a purine nucleotide for another purine nucleotide, or a pyrimidine nucleotide for another pyrimidine nucleotide. So we're replacing one purine already with another purine and pyrimidine nucleotide. A transversion is changing the type of nucleotide. So a purine is replaced by a pyrimidine, or a pyrimidine is replaced by a purine. So here's a diagram that shows you all possible transitions and transversions. So I want to make a comment here about the notation, because when you're determining, when you're ascertaining whether the base substitution that occurred is a transition or a transversion, it's essential that you look at comparable nucleotides. We're looking at nucleotide base pairs. So it's essential that you're making that comparison between the base that is the original and the one that is changed to. So here's this AT. If it makes it easier, draw these as you might expect to see them in the DNA, okay, as the double strand. And in that case, this A here is being changed to a C. A is what kind of a base? Purine, what kind of a base is C? Pyrimidine, and so this A to C, in the color beige here, is a transversion. So this would be like converting this to that, right? The A here is comparable to the C, vice versa. So when you're looking at these, if you're going to draw them this way, which is the easiest way to do it, make sure you're comparing the same positions. This A changed to a C. Here, this A is changed to a G. This A is a purine, it's being replaced by the other purine. And so that's a transition. So if you look at this, what would be the relative expected frequency from this random conversion, base substitutions of transitions versus transversions? Which one would you expect to be more frequent, just by random chance base substitutions during DNA replication? I heard transversions. Why? You're just counting up, right? Well, it turns out it's the other way around. Actually, transitions are more common. It's the most common base substitution. It's the most common point mutation. They're more common than transversions. And the only reason for that is that even though there are more opportunities, if you just look at changing one letter for another, there are fewer molecular chemical opportunities to convert a purine to a pyrimidine and vice versa. So transitions are actually more common. So let's talk first about transitions. The most common origin of transitions is from spontaneous change as a result of chemical isomers of bases. So what's a chemical isomer? All I have chemistry, it's a prerequisite. You don't need a lot of chemistry, you just need baby chemistry to recognize what an isomer is. What's an isomer?
It's a molecule with the same chemical composition but a different configuration.
Yep. It's the same chemical, it's the same atomic formula, right? But those atoms are arranged differently. And so it turns out that there are two primary isomers for every one, each of the bases. So this isomeric change, as one base changes from one isomeric form to the other, is referred to as tautomerism. This is true generally, that a conversion of that ability of one isomer to convert to another is referred to as tautomerism. That change in the structure of that molecular formula is referred to as a tautomeric shift. So we'll use these terms simply to talk about this, so you need to understand what that means. So let's look at the bases. If we look at each of the bases, adenine, cytosine, thymine, guanine, and you look at the structures on the left, The structures on the left are the more common isomeric forms of these bases in physiological conditions. So in your cells, the most common form of adenine is referred to as the amino isomer or tautomer of adenine. In order for adenine to undergo a tautomeric shift, the tautomeric shift in bases is the result of a proton that is off of so a hydrogen ion off of this nitrogen that is shifting from this nitrogen to this nitrogen. And when it does that, it forms the other isomeric tautomeric form of adenine, which is the imino form. So adenine is amino to imino, and there are isomeric forms for all of the bases. Cytosine, like adenine, has the normal physiological form is amino. Shift of the hydrogen from here to here causes it to become an imino. And guanine and thymine both have the most common physiological form being the keto form, where the hydrogen associated with this nitrogen moves to this oxygen moves to this oxygen, converting the keto into the eno forms. You need to know these. You don't need to know the structures. I'm not going to ask you to draw the structures. This is by way of helping you understand where these come from. But I also want you to notice the equilibrium arrows here. I said that these on the left were the most common, the more common of the two isomers physiologically. In fact, it's very, very rare that any of these bases spend any time in the unusual imino or enol form of the base. And it's a very short, this doesn't even represent how short that arrow pointing towards the unusual base is. But the most common one is this. So what? Why do we care? In shifting these hydrogens around, how do bases interact with each other? How do complementary bases interact? Hydrogen bonding. Shifting these hydrogens around alters the hydrogen bonding of the bases. So the two different tautomeric forms have different hydrogen bonding characteristics. All right, so just to remind you, I don't have to remind you because you all faced all of this and you're very, very familiar with it now. Cytosine, guanine, normally hydrogen bond, form 3 hydrogen bonds. Thymine, adenine form 2 hydrogen bonds. So we've got this purine with that pyrimidine, this purine with that pyrimidine. If in fact there's a shift, so if we shifted this hydrogen from this nitrogen to this nitrogen, the result is a change in the hydrogen bonding. So instead of adenine, instead of the amino adenine base pairing with which form of thymine? Which is the common one? The keto. The imino form of adenine is going to base pair with the amino form of cytosine. So the rare imino form of A is base pairing with the wrong pyrimidine. We haven't disrupted the purine pyrimidine base pairing, but the pairing is with the wrong, and we're wrong because that's not what you memorize is the base pairing, right? That's not what the normal base pairing is. And the same is true, you can look at the rare enol form of guanine, it should be base pairing with cytosine, it's base pairing with the wrong pyrimidine, thymine. And the same is true for the other bases. So each of these bases, if it shifts into the rare tautomeric form, it is going to, at that split second, it is going to favor base pairing with the wrong complementary base. Okay? This is easy to remember. A purine in its rare isomeric state is going to pair with the wrong pyrimidine. A pyrimidine in its rare isomeric state is going to pair with the wrong purine. Before we talk about how this comes about and how this tautomeric shift leads to these spontaneous transitions, I want to, an aside over here, an aside. You know about DNA structure. You know that Watson and Crick spent a lot of time and energy coming up with their hypothesis, their model for DNA structure, the double-stranded based on Chargaff's rule of the base pairing and so on. They had a terrible time initially. They were really, really stuck. And no amount of music was going to help them get through that brain freeze stop. And at some point, when they were doing all of this, they encountered some chemists came in to take a look at what they were doing. Because Hudson and Crick weren't by training hardcore chemists. And the chemists looked at them and said, I don't know what you're doing, you know? These, the G and T in nature, they're not the enol forms, they're present in keto forms. The light bulbs went off. Because of that altered hydrogen bonding, they were trying to make the double-stranded structure work with hydrogen bonding that didn't happen physiologically. They were following all of the rules for the physiological ionization of these different hydrogens and how the hydrogen bonding would, but it wasn't real life. And as soon as they learned that, they had that immense breakthrough that allowed them to put together the structure that fit the diameter and the consistency in the diameter and so on. Okay, just a little, chemistry is important. Okay, so where do these come from? Where does having this shift happen result in these kinds of base substitutions? Well, there are two places that this could happen. Most of these occur during DNA replication, right? When the DNA is replicating, and so it's adding nucleotides to a growing daughter strand in response to a template strand sequence. Well, during DNA replication, there are two places where a shift in tautomeric state can affect the incorporation of the nucleotide and result in a base substitution. One of those is it can happen to the template strand. So here is our DNA double helix. We're focused on this GC base pair. During DNA replication, we have this fork, the two single strands are opened up, and for some particular reason, no one knows why, that very, very rare occurrence of this gene, it switches from the keto form to the enol form. It undergoes a tautomeric shift. Normally, this gene would be a template nucleotide that would lead to the incorporation of a C, right, the way it was initially in the initial double helix. The incoming ENTP would be a DCTP, but now it's in this enol form, and that enol form doesn't hydrogen bond with cytosine, it hydrogen bonds with what? The other pyrimidine. And so what happens in that split second that gene has shifted over to the rare tautomer, it now, the ENTPs come in and the TTP is coming, the polymerase is coming along here saying, oh, this is the right base pair here because of the hydrogen bonding with that enol form of the gene. Now this is a rare event for that gene to shift, and it's going to shift right back, but the damage is already done. The polymerase has moved through and it has fixed this mismatch. This is one of the origins of mismatches during DNA replication. This G and T is a mismatch. And sometimes that will lead to enough of a bulge or blip or physical warning that the polymerase will be triggered to nuclease and proofread to get rid of the newly incorporated base. But if it doesn't, this will persist. Okay? If it happens in this, if the repair process of proofreading misses that mismatch, then this will be stuck here. So take this now, finished replication. It's finished replication, and this DNA molecule, the daughter molecule, has got its original template strand and the new daughter strand, so now it's got that mismatch. When this molecule goes through DNA replication, this separates. Notice, go back to the normal polymeric state. The G is going to have the C incorporated after it. And the T, since polymerase doesn't know any better, it says there's a T here, I'm going to put an A as the base pair complementary base, then this now has resulted in a result. It has resulted in a GC to AT transition because that G here was a purine and the A here is still a purine, so that's a transition. Okay? Questions on this? This is referred to as a template error because the error during DNA replication is on the incorporation of that template nucleotide, the reading of that template and putting in the incorporated ENTP, which is a completely normal nucleotide, it's just the wrong one. But the error happened in the template strand. Where's the other place that bases could shift from one tautomeric state to another? If it's not in the template. talk about DNA synthesis. Where else do you see nucleotides? If it's not in the template strand, where is the nucleotide? In the DNTPs, the precursors, right? They're nucleotides. They are just like any other nucleotides and having bases that can undergo tautomeric shifts. And so the other way that this base substitution transition can happen as a result of tautomeric shift during DNA replication is an incorporation error. That is, the mistake is happening, the error is happening as a result of the tautomeric shift of a DNTP that is getting incorporated into the growing daughter strand. So now we've got our double helix again. We're concentrating on this TA base pair. Here's the T, which is the template. Polymerase ought to put an A, a DATP, should be the precursor for that complementary base pairing. But instead, what roams around in its vicinity is the GTP, which has undergone a tautomeric shift, and that the G-enol TP has base pairing that is consistent not with the C, but with a T, the other pyrimidine. And so this shifted GTP can be incorporated into the site here, and that will result in, again, a mismatch. Here's the template T, the misincorporated G, but now it's a perfectly normal G, it's a normal keto G, and shift it back. Now when this replicates, it will produce the TA, which was the original molecule, and it will produce the CG. So that has resulted in a TA to CG, TC, pyrimidine to pyrimidine transition. So you see where those both can happen? Just random mistakes. Again, if this had been caught by proofreading, this might not have persisted. These mutations occur because they have slipped past any repair or proofreading. And so they have gotten away with that mistake. It wasn't fixed, it wasn't reversed, and so that mutation occurs. So if you want to think about this now, think about all the places where DNA replication is happening. Where are mutations going to have the greatest impact? What kinds of cells are going to be the most highly impactful for having this kind of mutation occur? It really is a gloomy day. Gloomy, sleepy day. Okay, so if this happens during replication in, let's say, in the liver, subset of cells, maybe still dividing in the liver, any big deal? Probably not, right? Out of all of these, just from this one replication followed by the second replication, only 25% of those cells, these are going to persist. They're going to keep getting flooded out by all of these, which are going to be wild type. So not a big deal, depending on where that is in a chromosome or gene. What about in a skin cell? Yeah, probably not. Where would it have an impact potentially? In the germ line, right? Because all it takes is for this mutation, that chromosome that carries this mutation, to be the one chromosome that is carried from the egg or sperm into the next generation. Every cell in that embryo has got that chromosome with that mutation. Yes, they've got also a normal chromosome with the normal gene, but this is the way that mutations get into a population and can be then inherited from one person to the next one generation. Okay? All right. So, transitions. Origin from, the most common origin from chemical isomers of bases. There's also another origin for transitions, and that is from alterations of bases called deamination. Sounds exactly like, it is exactly what it sounds like. D meaning taking away from, amine, amino group. The most common target for deamination is the base cytosine. Cytosine has an amino group, and if that amino group gets damaged or is some other way induced, we'll see it as a target for chemical mutagens, is lost, then cytosine that is lost its amino group, is hydrolyzed, is basically converted to uracil. Not a huge deal. because uracil is recognized by the cell, you shouldn't be in DNA. There's a repair process, a repair enzyme that recognizes uracil's out of place in that DNA of that genome. Uracil's supposed to be an RNA, right, not a DNA. And so there is a uracil enzyme that will convert this back into cytosine. So that's no big problem. The problem is that some of the cytosines in the cell are actually modified themselves. They're modified by having a methyl group. We'll talk about methylcytosine and the methylation of the DNA when we talk about regulating gene expression in eukaryotes, because there are some notable important processes where methylation of the DNA is critical for regulating gene expression. And the most common target for that methylation is cytosine. Well, if a cytosine, a methylcytosine is deaminated, it converts that methylcytosine into thymine. There is a repair process that can restore cytosines from thymine, but it's really, really, really inefficient. It's hard to recognize. Are you a real thymine here? Are you supposed to be there? Or are you a mistake from deamination of the methylcytosine? It's not really reliable. It's not accurate. And so many of these conversions from deamination of methylcytosine to thymine persist, and when they do, they are associated, the result is CG to TA transition, right? C pyrimidine to T pyrimidine transition. If you look at genomes, we have lost a lot of CG base pairs in certain regions that have been converted to TA base pairs for that reason. If there are regions that have been methylated cytosine. There's another base that is sometimes methylated, and I bring this up because it may tie some things together, and that's adenine. Deamination of adenine. Adenine also has an amino group, and if adenine loses its amino group, it converts it to hypoxanthine. Have you heard of hypoxanthine before? At least twice. Once in a fictitious practice problem set, but then I reminded you of it again when we talked about tRNAs. Anybody remember that? How is hypoxanthine related to tRNAs? Louder. Inosine. Inosine is a nucleotide that has hypoxanthine. How does it get there? Somebody asked me after class that day about how got these unusual bases in tRNA, how do they get incorporated during transcription? In fact, tRNA, those unusual bases, including inosine, are the products of RNA editing. And so after that tRNA is synthesized, what would be in that position of the anticodon that is destined to be an inosine is usually an A nucleotide. And the RNA editing removes the amino group from that A at that site in the tRNA, converting it to an inosine. So that's a normal deamination process. But if deamination of A elsewhere happens, then it converts that AT to a GC. It's not part of that RNA editing, but in the DNA, if the deaminated A persists, it results in an A purine to G purine transition. Does that make sense? Those are the two primary sources of transition. And as you can see, probably it's understandable why that makes transitions more likely to occur as a base substitution than conversion, because there are a lot of opportunities here, especially with those polymer shifts, for mismatches during DNA replication. So where do we get, what is the origin of the transversions? The most common source of transversion, in fact, transversions in general are not that well understood in terms of origin, but most transversions have resulted from a mistake, a process known as depurination. D, again, from, purine, purine. So it's the loss of a purine base, not the whole nucleotide. This is one of those rare instances where I make the distinction between a base and a nucleotide, even though there is literally a distinction, but we talk about base pair rather than nucleotide pair. The loss of a base. So in this, the target here is this guanine. It's the most common base that is lost through depurination. Guanine is a purine. If this linkage here which is the glycosyl linkage, the linkage that is attaching the base to the sugar, the purine deoxyribose sugar to the base, that linkage. If that linkage is cut or damaged or broken, it releases that purine, that guanine, from the polynucleotide polymer. It's not broken the phosphodiester bond. It has only broken that glycosyl linkage. So it's resulted in a strand of DNA here that has no base attached to the sugar. Not a big deal, probably. Sometimes it can lead to, depending on where it is and whether there's anything else in that region, it can lead to damage to the DNA, but most of the time these are perfectly fine. Well, what's the problem? What happens during DNA replication? Because it's that base that dictates the daughter strand synthesis. And so we lose that, and we've got that hole where there should have been a base, and because it was actually a purine that was lost, that site with the hole is referred to as an apurinic site. Pyrimidines can be lost as well, in which case this would be called an apyrimidinic site. Collectively, if we want to talk about a site that has lost either a purine or a pyrimidine, we would talk about it as an abasic site. We'll talk about abasic sites later. But this missing the purine, whether it's a purine or pyrimidine, if you don't have the information in the template that is going to dictate the daughter strand synthesis, it's a problem. So what I want you to do for next time is think about the problem of replicating. We start with a double strand. We've lost this gene, this purine is gone, the phosphodiester bonds are all intact, so we have an intact single strand. But now, this is going to replicate. Look at what happens here. There is no mechanism to replace that purine. There is a repair process to get rid of the whole region. We'll talk about that. But if this persists simply in DNA replication, there's no information there. The solution the cell has is, I don't know what it is, let's put an A in. I know what this is. Let's put an A in. There's nothing to hydrogen bond. I'm going to put an A in. Well, there's a problem here, right? Because the template strand had a G, which is a purine, but now the daughter strand has got a purine, and when take this through the rest of replication to follow through the consequence to see if you then get a transcript. Okay?